Hello, my name is Junwon Park and I'll be presenting on leveraging drone-based spectral analysis of plant moisture to combat wildfire risks. Our precious forests are suffering more and more because of global warming while they alleviate its impact. The annual average burned area in 2020 to 23 was 3 times that of 2010, showing the drastic impacts of global warming. The 2025 California wildfires also proved to be devastating, destroying 16,000 structures. Global warming is closely related to these phenomena as the gradual increase in temperatures and dry vegetation provides favorable conditions for wildfires. Then, what is being done to stop these disastrous wildfires that only get worse? Current methods of stopping wildfires focus mostly on early detection. Cameras and satellites alert firefighters of any wildfires, and they arrive at the scene to put out the fire. The problem is, cameras and satellites can only detect wildfires when they are clearly visible, meaning the fire would already be huge. So, in this research, I take an entirely different approach. Preventing these wildfires from happening in the first place would greatly reduce the impact and resources exhausted on stopping these wildfires. Dry vegetation is an essential component of these wildfires, as forest fire is significantly influenced by the thick moisture content of the vegetation that surrounds it, such as plants, leaves, and branches. So, we can predict areas with the highest odds of wildfire by finding areas with the most concentrated dry vegetation. Preventive measures such as controlled burning or artificial irrigation can then be focused on those areas to prevent wildfires from burning down forests. But how would we get plants to tell us how dry they are? Since plants and humans don't share the same language, we need to interpret plant signals. To do so, analyzing physical or biological differences of plants based on moisture level and conveying that to data is essential. When excited by light, plants emit fluorescence, and these fluorescence emissions vary with the biomolecular compound excited. Since the biomolecules produce changes as stress level changes, it also results in a shift in fluorescence intensities. This research hypothesizes that dryness can be quantified using fluorescence intensities and aims to distinguish healthy and dry leaves to apply findings to wildfire prevention. The purpose of this research is to develop a device that accurately measures plants' fluorescence based on their dryness levels to accurately quantify dryness based on fluorescence. By doing so, we will gain valuable data needed to accurately predict the odds of wildfire. Firstly, the fact that fluorescence changes based on moisture level had to be confirmed with the eye. So, Fluorogel, commonly used to observe the fluorescence of DNA samples with the eye, was used. In Fluorogel's dark box, an intense blue light was emitted to observe fluorescence, where red fluorescence was observed. The dark red box isolated all other light such that only the LED light and fluorescence would be present. Since small sensors are ideal for portable devices, the AS76 sensor was chosen and placed inside Fluorogel's dark box along with leaves to detect and quantify intensities of wavelengths at 6 different spectrums: R, S, T, U, V, and W. In certain experiments, a possibility of using a bandpass filter was tested out to concentrate light in the R and S spectrum as fluorescence could be disturbed by other sources of light. The code for the AS76 sensor is shown here and was run by Arduino and open source electronic software. A lot of different methods of measuring fluorescence were attempted to be used, such as fluorescence microscopes, a colorimeter through chlorophyll solutions, and a fluorescence detecting camera, but I concluded that measuring fluorescence with the AS76 sensor produces the most concrete and concise analysis. Diverse species of trees mean that fluorescence could differ between to some extent. Therefore, broad and pine leaves were studied in healthy, dry, and fallen states to account for seasonal and species variations. Since chlorophyll is the most researched fluorescent compound, chlorophyll solutions were also tested to isolate chlorophyl's fluorescence. Leaves and chlorophyll were excited by a blue light with a wavelength of 430 nanometers. Firstly, the relative wavelength differences of healthy, dried, and fallen leaves were measured through Fluorogel's camera system. Fluorogel converts fluorescence to different colors relative to their wavelength, so the colors shown are not the actual fluorescence of leaves, but relative fluorescence. Predominantly, healthy leaves show purple and blue, dry leaves show blue and green, and fallen leaves show red and yellow, establishing the foundation that fluorescence can differentiate between these leaves. Furthermore, the dominant wavelength leaves emit through fluorescence increases as the leaves dry out. To further analyze how fluorescence differs as moisture levels of leaves change, the AS76 sensor quantified intensities of fluorescence. Leaves were excited with blue light since chlorophyll and other compounds present in leaves produce fluorescence when excited by this wavelength. Fluorescence from leaves showed drastic differences between healthy leaves and dry leaves. healthy leaves produced high-intensity fluorescence in the T, S, and U spectrums for both the broad and pine leaves, with them sharing the same proportions. Meanwhile, dry leaves produce little fluorescence, clearly distinguishing between healthy and dry leaves using blue light. Fluorescence is an earlier indicator of dryness than visible light is, as both the healthy and dry leaves remain green. Fluorescence is superior to current camera systems that detect dryness based on color, as fluorescence gives more detailed analysis as well as signs of dryness that cannot be captured through visible light. Additionally, it's interesting to see how the exact same proportion of fluorescence were produced, as broad and pine leaves are completely different species. This suggests that there might be a universal trend in fluorescence in leaves across many different species, allowing for uniform analysis regardless of leaf species. Strong differences between healthy and dry chlorophyll solutions were expected as chlorophyll is established as one of the most dominant fluorescent compounds. However, fluorescence barely differed between healthy and dry chlorophyll solutions, suggesting otherwise. Healthy and dry leaves extracted chlorophylls have comparable fluorescence levels, contrasting with the healthy and dry leaves fluorescence. Additionally, healthy leaves have high T and U intensities, but for healthy leaves chlorophyll, these intensities are relatively low. These contradictions suggest that chlorophyll isn't the biomolecule primarily responsible for fluorescence in leaves. Rather, it is one of azulene, chlorophyll, phycocyanin, phycoerythrin, porphyrins, or quinones, as they produce fluorescence in the spectra studied. Among these, porphyrins are the most likely as they are both present in leaves and decrease in quantity when leaves dry, which would decrease fluorescence intensities for dry leaves. Sunlight contains white light, which contains blue light. These fractions of blue light could naturally trigger fluorescence, reducing costs for development. On the other hand, the filter could specify which wavelengths were measured, decreasing interference. It was attempted to measure fluorescence with white light and with a bandpass filter, but the white light did not really distinguish much between healthy and dry leaves, and the filter reduced fluorescence intensities by a lot, making the data prone to outside sources of light at that spectrum, such as sunlight. Therefore, it is best to measure fluorescence with blue light and without the filter. Fluorescence would be best measured from the sky as a large area can be measured simultaneously. So, field tests were conducted to confirm this possibility. The Portable Fluorescence Measuring Drone (PFMD), built with the AS763 sensor, Arduino mainboard, and 32 blue LEDs, successfully distinguished healthy, fallen, and dry leaves when tested outdoors. Unlike dry or fallen leaves, healthy leaves had SNT emissions of about 30, and their intensities differed by a factor of 3 on average. The entire PFMD system without the drone cost $60, so it is very cheap for what it could accomplish. Real forest environments, unlike lab conditions, have many different variables difficult for humans to account for, such as leaf types, arrangement, levels of dryness, distance from sensor to leaf, and wavelength intensities. To facilitate analysis, I needed AI technology with machine learning to predict dryness over a forest environment using fluorescence values. Since both broad and pine leaves show similar proportions in fluorescence, I developed a model that can analyze dryness regardless of the species of leaves measured. 9 different types of leaves were selected and assigned a dried score based on how well they dried. The 9 leaves were arranged in 31 different arrangements to simulate location variables. The total dried score was summed up and divided by the surface area of leaves to get dried score per area, a metric that quantifies how much the leaves had dried. AI predicted the dried score per area using different variables. Through the random forest regression model, the AI was trained by predicting the dry score per area with fluorescence intensities and comparing it with the actual dry score per area. Models solely using each of the 3 intense spectrum data and a model with 39 different variables were developed. The 3 spectrum model showed similarly low mean absolute errors and mean squared errors, with high R-squared values, indicating dry score per area can be accurately predicted when using any of these spectrums. However, among these 3, the U values showed the highest R-squared value and feature importance in the model involving all features, meaning it contributed the most to the accuracy. The U spectrum values are the most promising for identifying dry vegetation in forests using dry score per area. Predictions align closely to actual dry score per area values. Since the AI model was trained with a sensor placed 30 centimeters above the leaves arranged in a 20 x 20 centimeter square, a ratio of 3 to 2 from height of drone to width measured would be ideal for measuring fluorescence. For example, when measuring an area of 200 times 200 meters, the drone would be 300 meters high in the sky. The AI would then predict odds of wildfire in each area. Since current efforts to fight wildfires mostly involve stopping wildfires rather than preventing them, this research focused on using fluorescence to analyze the dryness of plants to prevent wildfires. The PFMD with the AI model can predict dry score per forest area with fluorescence and map out the dried areas. Preventive measures such as artificial irrigation and control burning will be implemented in the driest areas to prevent wildfires and save water. For the future, an upgraded AI model can combine external data on temperature and precipitation to find correlations between weather and dry score per area. Wind data can also be analyzed to predict where wildfires will spread based on the spread of dry vegetation. Some limitations of this research include the lack of hardware developed and field testing of the AI model. However, once the prototype is complete and is applied to forests, the PFND will output promising results on humanity's fight against wildfires.